This video describes the Caltech Clover Center's protocols for producing engineered systemic AAVs. There are many reasons to want to make your own AAV, but some specific equipment and infrastructure is required, including a BSL-2 cell culture facility, an ultracentrifuge, and a qPCR machine. These protocols are described in detail in our paper in Nature Protocols in 2019. The entire process takes about 8 days, starting with transfection on day 0 here. HEC293T cells are used, and normal care and considerations should be maintained in their passaging. During transfection, each cell requires 3 separate plasmids, one encoding the recombinant AAV's genome, another encoding the capsid proteins, and a third encoding additional proteins necessary for replication. Importantly, the ITRs of the AAV genome are capable of recombining out of the plasmid while the plasmid is being prepared in bacterial cells. Therefore, for each new plasmid prep, the integrity of the ITRs must be reconfirmed. This is usually done by restriction digest. Transfection is fairly standard using PEI. You'll want to drip the transfection DNA mixture onto the cells and then swirl the plate so that all cells are equally receiving the mixture. After 12 to 24 hours, you'll want to remove the media and replace it with fresh, as PEI is cytotoxic and will reduce yield. About 72 hours after transfection, you should be able to see transgene expression, and most AAV genomes contain a fluorescent protein for this purpose. On day 3, you'll collect the media and replace it with fresh. This media will contain 10 to 20% of the expected viral yield, and the cells will require fresh media to continue healthily producing virus through the remainder of the protocol. Final harvest happens on the 5th day after transfection, and you'll scrape the cells toward you to liberate them from the plate, and then collect them and centrifuge the media and the cells. Supernatant will be combined with that harvested at day 3, and the cell pellet will then need to be lysed. One important note that separates our engineered AAVs from the natural serotypes is that cell lysis should not be done by freeze-thaw or sonication or cavitation. These capsids are a little more sensitive, and osmotic stress works best for us. We use a high salt solution, which then can be paired with the salt-activated nuclease so that the lysate is not too viscous to work with. Virus that is in the supernatant is PEG precipitated and then resuspended in its own SAN digestion buffer and salt-activated nuclease. These are combined, spun, and then the supernatant there is taken forward for purification. The virus is purified through an iodoxanol density gradient. First, you would put the 15% iodoxanol down, and then you would stepwise add additional layers at higher density underneath it. So you'd take a 2 ml pipette containing 3 mL of iodoxanol, touch it to the bottom, lift ever so gently, and slowly place the denser layer underneath. When getting to the end of the volume in a pipette, you want to be very careful not to release air bubbles. And so a small volume can be left in the tip, and as you raise the pipette slowly, it will move out. it will take 2 full 2 ml pipettes in order to deliver the full volume of each layer. Once all the layers have been added, you're then ready to add the supernatant containing your virus in the sand digestion buffer. You would then fill the whole tube up to the top with additional sand buffer, cap it, and then measure the weight of the fully capped and spacered capsule such that they can be properly balanced in the ultracentrifuge. After ultracentrifugation, you must extract the virus from the 40 and 60% interface of iodoxanol, where it equilibrates during the spin. To do this, you puncture the capsule with a needle right below the interface, and then you'll collect about 4 to 5 mils of this virus. Most of the full capsid resides right at the interface where it looks orange-yellow, and as you collect toward the precipitated protein layer, you will get greater yield, but you will also lower your full to empty ratio. And it's very important to rotate the bevel away from the protein layer as you approach it so that you do not accidentally take up more protein. Once you've extracted the volume of the virus, you'll then recap the tube, remove your syringe, and then place the iodoxanol containing the virus that you've extracted underneath a layer of PBS in a syringe connected to a filter. The top layer of PBS will then be used to wash the syringe and the filter as you push the material into an Amicon tube. Once in the tube, you'll need to mix the material well so that the iodoxanol is evenly diffused across the PBS volume, as this will speed buffer exchanging until you have diluted the mixture into entirely PBS. Finally, after purifying, you'll need to titer your virus. To do this, in broad strokes, you first digest DNA, which is not packaged into the viral capsid, by adding DNase 1. Once this is inactivated, you then add proteinase K to digest the capsid protein and reveal the viral genome that had been protected. Finally, you would do a qPCR on the viral DNA against a standard. It's very important that the qPCR amplicon is validated and is not within the ITRs. We have found that trying to qPCR off of the ITRs leads to inaccurate quantitation. There's a number of references that have gone into this video, and if you have any questions, feel free to contact us at Clover.